can skip something for sure. So I am Flavia Villani. I come from Italy, and I received a bachelor degree in biological science, then a master degree in medical biotechnology, and I started to learn about bioinformatics. And then I done 1 year as a graduate research fellow in this lab, and I started to learn about Pangenome. That is my research field now. And then I started the PhD. This is my second year, so I'm finishing my second year. So the thing is that I work with the Pangenome. I don't know if you know. Okay, I can try to explain to you. A Pangenome is a set of sequence altogether. So we don't consider a sequence against a reference genome, but we consider all sequences together. And this is an example of Pangenome. It's not related to a graph, so I don't know if you see sometimes that. This is an example of Pangenome. So I can skip this because it's too hard. So why we need a Pangenome? Because this is the standard genomic approach. So all sequences, as you can see, are aligned against the reference genome. And also, if you have a new one sequence, also this one is aligned against the reference genome. Instead, with the Pangenome, all sequences are aligned against each other. So we don't need a reference genome. And also, when we have a new sequence too, also this one is aligned against the others. So what is the aim? The aim is that in this way, we are able to capture all possible relationship between sequences, and we are able to identify structural variation in particular and so on. So the thing is that I contribute to build this tool. This tool is useful to build the Pangenome. The first step is the alignment, so all sequences are aligned against each other. The second step, this alignment is induced in a graph, and this is a representation of a Pangenome. So this is the representation. Each line corresponds to a sequence that you have, and the variation are represented by vertical lines, as you can see here. So we apply this tool with human. In fact, in human, as you know, I don't know, as you know, we have the first telomere to telomere genome assembly, and we use this to build the Pangenome from human. This is only collaboration that I done. But my principal aim is working with model organisms, in particular with mice. And I started to work a little with VixD mice, and the thing that I done, the problem is in this thing is that we have short reads technology, 10x linked technology, so it's not simple to rebuild the Pangenome and done some biological stuff. But I tried to do that. This is an example of Pangenome of chromosome 19. And as you can see here, this is really good, but the problem is here near to the centromeric region, because we are not able to rebuild this portion because the reads that we are using are too short, so it's not possible to do that.
Okay, let's go back to that slide.
Yeah.
So, this is, I understand.
Yeah.
At least I know what the colors mean. Would it be helpful to add another seven rows?
7 rows? What do you need? No, because
you have 148 strings, right? Would it be a good thing to add another dozen strings to that?
No, because the problem is here is the technology.
I thought no was the correct answer. Now let me ask the next question. Would it be helpful? Would it be harmful to remove 12 strains from that?
No.
Would not be harmful.
I don't think it's.
I agree with you. Would it be harmful to remove 100 strains from that?
No.
Okay.
It depends.
Would it be harmful to have 10 strains and only 10 of your 148? Would that? I can see why having 10 randomly chosen strains might give you lots of information, but I'm not sure why adding the next 138 puts you in a position to do good stuff.
The problem here is that I started with the only 2 strains.
Yes.
To test the tool and see what happened. And then I tried to rebuild to use all the strains that I have. But the problem here are the data that we have. So if I use 2.
Why are you doing this?
Why?
Why is this good?
Why it's good?
Yeah.
With the pangenome is
Yeah.
good. It's good because we have all sequences against each other. We use all together.
You don't have all sequences. I can find you another couple hundred mice that aren't on that list. So you don't have all the sequences. You have some of the sequences.
Flavia, what you forgot to say is this is all 148 here are BXGs, right?
Mhm.
So you really only have 2 genomes.
Right. So there.
Yeah,
that's
but
even
if
worse.
no, sorry, but if you have if you have two strains, for the parental, you can use these two together,
Yes.
and you don't have a reference genome,
Yes, I see that.
so you can see something
Yes, you
else.
can definitely make a big matrix with all of those things lined
All
up.
the
I'm trying to ask
different
why would you want to do that?
I want to do that because my aim for now, my last aim is to have a new third generation sequencing, to have telomere to telomere assembly from the parental strain and for F1 generation. And I want to try to see if I am able to use the pangenome markers to improve the AQTL that we have already on GeneTwork, and also to study methylation around the centromeric region, because as you can see here, we don't know anything about the centromeric region. So if we will have good data, we can go near to the centromeric and study methylation, transposable elements, and so on. I'm sure that I can't do all of this, but this is
So this is what they call formative feedback. Getting questions like this is good, but you haven't given an efficient answer. And it's very easy to just say genome to phenome, genomes to phenomes. You've got 148 variants on a theme, right? It's like, Bach version 1 versus Goldberg version 148. And now the question is, does it put the listener to sleep or does it make them vigorous? So genome phenome is where you want to go here.
Yeah, the first thing that
And you
I
definitely
can do.
have to be clear that this is not really 148 strains, because if you say that to anybody, they'll think, where did you get your strains? Are they from Jackson Laboratory?
Yeah. So yeah, this is so I think I can stop here because we talked about